Without further ado, I would like to start the session by inviting Dr. Gintaras Brazauskas, who is not only a director of Lithuanian Research Centre for Agriculture and Forestry, but as well he's an actively conducting research in the Plant Genetics and Physiology Lab. Gintaras' research interests include plant adaptation, gene identification and expression analysis, as well as DNA marker development and application in cereal and perennial grass. Gintaras, I hope you're all set to go.
Yes, thank you. Let me share my presentation. I hope it's nicely on. Thanks. Okay, so I'm going to present the project which we just started from the beginning of this year. So not so much results to present. I'll present mainly the idea, the partners, the rationale behind this project. So the short title for the project is Noble Wheat and deals with breeding tools we are going to develop for the spring wheat breeding in the Nordic Baltic region. So we have 5 partners of the project. You have the logos of all the partners, all the institutions. So starting from us, then also Norwegian University of Life Sciences, then Institute of Agricultural Resources and Economics from Latvia, Estonian Crop Research Institute, and University of Tartu from Estonia. We also have an external expert from Swedish University of Agricultural Sciences. So 5 institutions in total. And we are all set to develop the breeding tools for this Nordic Baltic region. So you have 8 countries in this region, 3 Baltic states and 5 Nordic countries. So Northern European region, where the wheat production is quite important. So you see a list of countries. Iceland is out of the list. They are not cereal producers at the moment. So the list of the countries, all countries sorted according to the cultivation area of wheat, which in total is almost 3 million hectares. And then the harvest, these figures are from the last year from the Eurostat. So 16.5 million tons of wheat was produced in this region last year. And the yield is quite variable, you see. Depends on the location of the country and the practices applied. So in total, it is 13% of EU common wheat production area, and which produces 14% of the total wheat production. And quite a substantial part of those wheat is exported within or outside EU. So especially the Baltic states are quite active in this, and 6 million tons of the total 16 million are getting exported. So wheat is an important commodity in this region, and with the climate predictions, we have a quite positive prediction for the wheat in this region. The temperatures might rise, or most probably will rise, and so we'll have beneficial conditions for the expansion of the cereals and other crops in this region. But the challenges will come as well together. New diseases, insects, and also, while the temperature will change, the photo period will stay the same. So new idiotypes of the plants, of the varieties, will be needed, and that's where the plant breeding will come in to provide those genotypes. Another pressure is, of course, the EU Green Deal strategies, farm to fork specifically. It was mentioned a lot of times during this meeting and other meetings. So agriculture will face less input, and if you want to stay as productive as we are now, so new technologies, new inventions will have to be provided for the wheat production. And we have to keep in mind that farm to fork strategy is not only about the production, it's also about the processing, sustainable consumption, and also food loss and waste prevention. So every one of us has a role in it, and not only the producers. But however, if you want to have affordable, good quality food and sufficient amounts of food, so we have to find inventive ways to maintain the production by maintaining the same production area. So new tools will be needed, and there is already quite a list of the tools available to the wheat breeders. And first of all, of course, the genomic tools is a complicated diagram, of course, but so is the wheat genome. It is 5 times the size of the human genome, and it took almost 20 years to sequence the wheat genome. But good news, a couple of years ago, the wheat genome was sequenced, is now publicly available, so all the breeders can exploit this tool now. Genomic selection is a specific technique, the selection based on the genomic estimations. So it is also already widely used in the breeding process. And targeted mutagenesis is also a novel tool developed just a couple of years ago. Genome editing, I hope most of you heard about it. So those techniques are coming in, and especially the new communications from European Commission, the new study, there is some hope that it will become the adopted technique in the European Union as well. Phenomics is another group of tools available, and those are different techniques to phenotype the plants, and there are the various examples from very high throughput, high-tech technologies, like you see one from the Rothamsted research in UK on the top left picture, one from ETH Zurich in Switzerland on the bottom right picture, and various different other techniques. And there are also simpler but also very robust techniques like this PhenoCart. So a lot of available tools to process images, to acquire real-time imaging of the plants on the field or in artificial conditions. And once you get this great amount of data, both genomics and phenomics, of course, bioinformatics has to come in, and artificial intelligence is also one of the techniques which is more and more widely applied in plant breeding and also wheat breeding as well. So let's come back to the project. We have 5 work packages. I'll go step by step through those, so no need to read them all now. And let's start with the work package 1. So we have collected a core germplasm of 300 genotypes of spring wheat genotypes. So we have 4 breeders in this project, so every breeder provided 75 accessions from their collections. We have multiplied those, and we have established evaluations or field experiments at 4 locations. You have those marked in 3 Baltic states and also in Norway. So we are going to phenotype those in over 3 seasons, mostly for yield and other agronomic traits, and disease resistance is one of the traits which we are looking for, and many other traits as well. So there is the first picture from this year's trial. Not so much to see. It is just 2 weeks after sowing, if I am correct. So not so much vegetation, but you already see the imaging tools we are applying in this process. We'll come back to that picture later. So the second work package is implementation of the plant phenotyping tools in Baltic wheat breeding. So we use 2 types of phenotyping platforms, so proximal sensing vehicles or phenomobiles. And this one picture is from Norway, from our partner, Norwegian University of Life Sciences. They use that Saga Robotics produced autonomous device to drive through the field and do the phenotyping. While the Baltic partners, they are producing simpler techniques, but also very efficient. This is already from our side. The PhenoCard, where you could apply different cameras to collect the phenotypic data. And also UAVs, so drones, those are also more and more applied in agriculture, and especially with this DJI Phantom 4 multispectral cameras, you can do a lot in a quick and efficient way. So all those techniques will be used, and lots of data collected, or already being collected, and then the analysis will be one of the tasks of this work package. So a bit more detail. So the PhenoCard, we have applied that already last year in a winter wheat, and you can see there's two cameras attached to the device. And those two cameras take simultaneously two separate pictures. One is RGB, or ordinary digital picture. And the other camera is slightly modified, so it takes near-infrared pictures. And by combining those pictures, you can estimate different vegetation indices. And one of the most popular is NDVI, or normalized difference vegetation index, which provides information on how the plants feel. So this is a very important value to go through the collections of various genotypes and see how the plants feel. The remote sensing phenotype, we already started this year phenotyping, and this is handy, especially this year. We have an extremely rainy May, double the amount of the rain precipitation. So it's not possible to go to the PhenoCard on the field, but you can easily fly the drone. So you saw already this picture, that's RGB picture. Difficult to see the vegetation emerging already. But if you apply NDVI filter, you calculate NDVI indices, you can clearly see that we already have a good germination of the plots, and you also could see some differences between genotypes, but it's of course a long way to process the images and extract the meaningful results from that. Move on, so that was phenomics part of the project. We also will do the genomics of those 300 accessions. So we're going to extract DNA from those genotypes and genotype those. And then market rate association analysis will be the main target to identify the beneficial alleles for the traits we're going to phenotype. And also genomic selection, that's a new tool, slightly different from market rate association, but very handy in breeding. So we're going to estimate breeding values of those genotypes as well. A specific work package 4 is conducted by the Tartu University group, a group from Hannes Kollist. They are very experienced in the plant gas exchange measurements, and they have developed that equipment for the indoor and also outdoor measurement of the plant gas exchange. And that's going to provide the information on nitrogen use efficiency and water use efficiency of 16 selected genotypes in this project. We also conducting the separate field experiment with the different nitrogen applications to see the nitrogen use efficiency of those 16 selected genotypes, and we'll do genomics on that as well to see how could we improve the nitrogen and water use efficiency through wheat breeding. And the last work package is work package 5 on knowledge exchange and network enrichment. So we're gonna organize annual training schools and project meetings, and also expand our network, which is quite extensive already. So there are different initiatives like Baltic Wheat. We have a separate network on wheat in the Baltic Nordic area. Then NPPN is a Nordic plant phenotyping network funded through the Nordgene. Nordplan is also plant science network among the Nordic organizations, and also the European Cooperative Program on Plant Genetic Resources, so-called European Evaluation Networks. So most of the partners are involved in those as well. So there is quite already substantial network, but we're going to expand through this project, and the ultimate task is to have those tools available for the future weed breeding, and also to expand our participation in the future projects. So that was a brief introduction. Thank you.
Thank you very much, Gintaras. Yes, we are working towards greener, competitive, and more inclusive Europe. And can I just ask you, out of interest, so you mentioned artificial intelligence and some other elements, technologies you're using, and the focus of this research is clearly Baltic region. Do you think some of the methodology that you will be using can be applied to other regions for exactly the same purpose?
Sure. Well, our specific target is this region and the future climates. So we are trying to predict the future climate and develop the best genotype for this climate. But of course, the knowledge we generate, or the functional mechanism behind that those are universal and could be applied in other regions as well.
Thank you. And we have a question as well from Mohammed. Are you considering to develop seeds or plants that could be disease resistant and also different stresses resistant in order to reduce the use of chemical pesticides and chemical fertilizers, which can lead to achieve zero pollution, sustainable agriculture, and then greener global world?
Yeah, sure. So I already mentioned that disease resistance is one of the targets for those gene trait associations. And the experiment of nitrogen use efficiency, that's clearly has a clear aim to develop genotypes which use nitrogen more efficiently, so less nitrogen would be needed to be applied.
Thank you. And the last question, so what happens when the project finishes? Are you going to apply for further funding to develop this idea further? Because as we know, project finishes, and then what's next?
I would say if you wait until the project finishes, then it's too late. You have to get ready before the project finishes. So that it's really challenging to fund the research continuously, but that's all we have to try. And I think by having events like this, and also by building networks, that's the way to go forward.
Yeah, absolutely. So if you have any further questions, please put them to the chat for Dr. Brazauskas. Otherwise, get in touch with him and his network if you want to collaborate. And now I would like to invite Dr. Žydrė Kadžiulienė, Chief Researcher at Department of Plant Nutrition and Agroecology, who has an interest in agronomy, grasslands, agroecology, crop management, ecology, and environment. Žydrė, over to you.
Yes. Good afternoon, everyone. Dear participants, dear organizers, thanks for chance to share our results, our small input in legume research in different projects. And in nowadays, in EU, attention was particularly given to Green Deal and various important strategies mentioned today many times, whose main objectives related to climate mitigation, to biodiversity preservation, environmental protection, etc. And food legumes and other legumes in production value chains, as many of you know, can play an important role by delivering multiplier services in line with sustainability principles. Legumes play central roles at food system level and production system level and cropping system level. and therefore the legumes production is viewed as promising option to mitigate climate change, providing nitrogen to soil and limiting external use of inputs, and increase in the proportion of leguminous crops on arable lands is crucial in order to decrease both pesticide use and nitrogen fertilization. However, in practice, at that moment, as the Lithuanian case, the use of legumes is still low, and a little more than 7% in cropping systems, and different reasons in different countries exist, but some is the same, and exist also some barriers as yield variability, weediness, disease and pests, a large phytosanitary break, and difficulties in harvest, and also a lack of knowledge. Until now, researchers paid attention on legumes, and many EU and national projects run related legumes, trying to find and propose solutions for farmers, consumers and politics. And one of the project, Leg Value project, EU project, Leg Value project is fostering sustainable legume-based farming systems and agri-feed and food chains in the EU, which participated 24 institutions from 11 countries, and LAMS is one of the participants. This project is quite big with few objectives and many tasks, and expected impacts. This year is finishing year. Expected impacts on sustainable legume-based cropping development and increased competitiveness and reduce environmental impacts, and different methods were included, and literature analysis, networks of farmers, interview, case studies, and different questions in legumes value chain. And due to literature analysis, as you can see in the slides, still exists a lack scientific information, and this project devoted to improve this knowledge shortages. We in this project focus on legumes in cropping systems and communication with farmers, and focus on research also on peas, fava beans, but also legumes less studied and cultivated in northern countries, but interested by farmers. As you know, soybean represents the most important and cultivated legume in the world, but areas in Europe are constrained by climatic factors. But climate change, new varieties comes, better technological abilities, and increase of demand for local resources provide significant reason to introduce soybean in northern countries. And this research, which you see in slide, examine 2 sowing times, row spacings, and inoculation of soybean, and the interaction of all these factors on soybean productivity in an organic farming system. Yield varied from low 600 kilograms per hectare to 3,000 kilograms per hectare in response to management factors, but this study suggests that regular soybean development could be maintained in organically managed locations above present northern soybean distributional region, but its development, productivity, and production quality significantly depends on practices and, of course, meteorological conditions, as often it happens. Legumes for more efficient use of nitrogen in crop rotation, especially in organic farming, also very important. And the pre-crop value of different types of legumes varies, but all legumes improve crop rotation productivity and supplement with nitrogen, and the greatest effect on subsequently grown spring wheat was exerted by the mixture of spring barley and red clover, and spring wheat produced higher yield after all legumes than after sole cereals. But in order to improve nitrogen balance in spring cereals, soybeans are very useful as pre-crop, but at present it is more useful to grow red clover. The results of subsequent crops, of course, depending on nitrogen content incorporated in soils and legume species, as you can see, different and on right slide, different legume species, and as usually after forage legumes, nitrogen content higher than after grain legumes. So response of crops also is on winter wheat, for example, is different. Red clover and organic fertilizers for subsequent crops in stockless organic farming also need attention, and in nowadays agriculture, the number of farmers who operating stockless cropping system has been on increase in many countries, and in such cropping systems, legumes could serve as a management tool to mitigate reliance on external inputs. Also important aspect that based solely on legume fixed nitrogen is not easy to meet the nitrogen need for cereal crops at the right time. Therefore, it's important to find measures contributing to grain yield and quality. And effect of legumes and manure pellets on spring cereals productivity were investigated on different soils. And as you can see, this many figures table, incorporation of red clover biomass into cropping system had a significantly positive effect on yield of wheat. But cattle manure pellets not was so significant on wheat, spring wheat, but cattle manure pellets used for cultivation of Watson's spring wheat had positive impact on barley yield, and the results confirm the findings of other researchers that to maintain the soil fertility with legume nitrogen and with low input organic fertilizers in organic stockless crop management is not very easy. We have to search other measures, and legumes and plant-based materials and organic fertilizers also are the measure for supporting nutrient cycling in stockless cropping systems. However, most utilization ways of legume biomass are limited in time, and which particularly difficult to meet the needs of the main crops for nitrogen, especially in spring cereals. So assessment of effects plant-based fertilizers on subsequent crop productivity potential is especially important in organic farming, and we examined the effect of plant-based fertilizers and silage and compost fertilizers on spring wheat and spring barley productivity and nitrogen accumulation in cereal grains and the stockless organic aerobic systems. Field studies were conducted on loam and on clay loam soils during 2 years, and we get that organic materials and could have effect, and most of the nitrogen yield was accumulated after application of green or and silage red clover biomass and granulated cattle manure. The use of plant-based organic materials and granulated cattle manure affected also on spring cereals productivity, and the productivity and quality of spring cereals in stockless organic cropping systems can be improved by use red clover incorporation in combination with as management factors. We have also a project, institutional and or national project on smart legume or enhancement of the multifunctional properties of legumes in feed and food values chains. And in this project, we focused on new varieties of peas and agronomical and technological properties of lentils and chickpeas. The legumes as and soya not very cultivated in Nordic countries and in Lithuania also. So we get some results, and lentils could be a potential future crop in Europe or in Lithuania. But study shows that grain yield is still low. It could be obtain some yield, but need more research and more development innovations. And we have in this project provided 2 new pea varieties. We have pea breeding in our institution, and here you can see some results, and new varieties Agla and Glinda, and in preliminary experiments they are yielded than oldest variety Jura, and we expected to provide some new recommendations specific for each of variety on the end of this project. And again, legumes, green legumes, very important, but we can't forget forage legumes, which are effective in case of enhancement of biodiversity, and especially in reducing mineral fertilizer usage. And as you can see in this last slide, that multi-mixture swaths of legume-based swaths are more yielded than monoculture swaths, even fertilized with 150 kilograms of nitrogen per hectare per season. And we must to explore these effects and to cultivate more multifunctional swaths in our lakes and grasslands. So it is all what I wanted today to say, and as conclusion, of course, need more legumes and need more new suggestions and new research on legumes. And we expect that legumes in future farming and food value chain will be more and more. Thank you for your attention.
And thank you very much, Jidre, for sharing your research insights, findings, and the projects that you're working on. I have a question for you. You mentioned about lack of knowledge when we talk about the legumes and yielding of them. And that is it a lack of knowledge on the side of the scientists, or on the side of the farmers? Where that lack of knowledge sits?
I think it's both. Not scientists knows everything also, and farmers also. They have some farmers are very smart and use very innovative technologies, but some farmers need some new, not new maybe, and not very new, but knowledges to improve.
And is the other legumes less resistant to the diseases and climate change, like, for example, wheat that we just heard from Ginteras before?
Legumes also have problems with disease and pests, and when we grow less areas of legumes, we haven't so much problems with disease and pests, but when we will cultivate it more and more, it will come these problems. So it needs to work. Also, it's work for plant breeders also.
So it's kind of going back to the same context, isn't it? Everybody was talking about farm to fork. So we have to inform the society that legumes are great, they're healthy, they're grown organic. So then that would increase probably demand from the farmers to produce more. And then we need the breeders and other scientists to get involved in order to yield a bigger crop.
We need all us involvement to this goals or objectives to get in future.
And have a diversity of crops as well, which is very important.
Yes.
Thank you very much, Jidre. You have a question in the chat, and I will leave you to answer it as we are moving on with our presentations. And I would like to invite Dr. Efi Alexopoulou, a researcher at Biomass Department in Center for Renewable Energy Sources and Saving, otherwise known as CRESS. And Efi is not only a researcher as well, she's an amazingly successful coordinator of many groundbreaking EU-funded projects, such as Fiber, Magic, Panacea, 4F, Crops, to name the few. Probably there's dozens more that I have missed off, but I'm sure you can tell all about it yourself, and the floor is yours. Welcome. We can't hear you. Oh, would you able to unmute yourself? We still can't hear you and can't see you yet. We can see the presentation, but we can't hear you. Okay, so...
Can you hear me?
Yes, now we can.
Now?
Yes.
Okay, because it seems still unmuted. Okay, can you hear me?
Yes, we can.
Okay, first of all, I would like to thank you for your kind invitation to participate in this very successful conference. I enjoyed a lot so much. So far, it looks very interesting for me. So I would like to present the role of non-food crops in agriculture in terms of biodiversity. And this presentation is based on two European projects, Panacea and Magic. Okay, so let's say, do you want full screen?
I'm sorry, could I just ask you? Yes, full screen, thank you.
Okay, so, but what are the non-food crops? The non-food crops can be even food crops, but the term non-food is connected with the end use. When we produce them to take bio-based materials, biopolymers, bioenergy. So we adapt this term for Panacea project that just completed, and Lithuania was one of the partners, your institute, for instance, and your participation was very, very successful. And I would like to thank you, Vita, for the kind invitation for this. So what are the main categories of non-food crops? We group it to oilseeds, let's say like camelina, rapeseed, sunflower, to fiber crops like flax, kenaf, industrial hemp. We have the perennial grasses, like giant red miscanthus, the woody species like willow and poplar, and finally carbohydrate crops like sorghum and sugar beets. So we have to think which are the end use of these products, of these crops. We cultivate them either to take energy, material for chemistry, for nutrition, or pharmaceutical uses. And here you can see how much biomass volume we want and which is the end price. So for bioenergy, we need a large amount of biomass, but the price is low. On the contrary, when we cultivate aromatic crops, specialty crops, we don't need large area, but the price is very high.
I'm really sorry, Effie, we can't see the full screen, and we're still on your second slide where you introduce non-food crops, the description.
Now I'm in the fourth slide.
We can see only the second. It's okay, but could you, I wonder if
you want to try to share the presentation that I gave you?
Yes, we're going to ask Vita if she can share maybe the presentation.
Yes.
Because you're talking so interestingly, we don't want to miss any of the important points.
Okay, if you could stop sharing, then I could share my screen.
It happens again with, I think, Teams, so
that's fine. The Teams had an update, I think, so a couple of days ago, so honestly, okay. You're not on your own.
So, can you see, did I stop sharing my screen?
Not yet. Now you did, yes, thank you.
Okay.
Now we're going to just wait for the Vita to share hers, and perfect. It's always good to have a backup, so you can't. Yes. It was a premonition that something might happen.
That's why I sent it this morning.
Brilliant.
You can see now the screen, yes?
Yes, perfectly okay. So we can go to number 3 slide. Okay, and now to number 4, where I'm going to explain. And now in number 5, I did that. That is the connection between the volume and the price in terms of non-food crops, and then go to the next slide that was the base of Panacea project, and this came the idea. We saw that in Europe there are many successful cases with non-food crops here and there, and we thought that the farmers need guidance, so we established this thematic network to promote non-food crops in European agriculture and to design the roadmap how to establish non-food in European agriculture. So we had 10 countries, and we saw that most of them, they have common crops. If we go to the next slide. Okay, we have to think always that we want to keep biodiversity. So we need to have healthy fields. We want not to harm the insects. We want to have pollination, and we want to increase biomass yields. But how non-food crops connected with them? Usually, non-food crops are crops that don't need a lot of inputs in their cultivation. They can be grown in rotation with conventional crops, and moreover, can be grown on marginal land, as per annual or perennial. So all of these, we can use it, and we can increase biodiversity. Keep in mind that the CAP after 2020 is going to put a lot of emphasis on some specific non-food crops because they increase biodiversity, and RED 2, that is the directive for biofuels, put emphasis on growing crops and to increase soil carbon and at the same time increase biodiversity. So all of these are connected. So we can go to the next slide. And I'm going to present some examples. So in Panacea, we saw that many partners, they were interested, many countries on Camelina, and some of them on Ethiopian mustard. But what I saw, Camelina was so interesting for the majority of the crop, on the partners. Camelina has very short production cycle. This is an asset. So we can grow in double cropping, and in 1 year the farmer can take 2 crops. They can increase the income, and at the same time Camelina does not have a lot of enemies and is a low input crop. It is the same with Ethiopian mustard, and both of them, they provide feedstock for bio-based products and bioenergy like biofuels. If we go to next, in the next we can see miscanthus and switchgrass, that both of them, they are perennial grasses. If we go through in European level, we can see that miscanthus is being cultivated in a total area of 25,000 hectares, and is used a lot for phytoremediation. Phytoremediation was presented in session 1. So miscanthus has been selected in many areas for this reason. But if we see this study, we can see that compared to corn, switchgrass and miscanthus improve all in terms of all elements in terms of biodiversity. So we can go to the next. Here is an example for another project, BeCool. BeCool is a project on advanced biofuels. It's a project between Europe and Brazil, where we are testing lignocellulosic crops, non-food, in rotation with maize and corn. And we proposed 6 rotations where the aim is to increase the biomass yield from the same area and not to jeopardize the production of the food crops, maize and corn. So we saw after 3 years that not only we didn't receive less biomass, less production in terms of corn and yield and wheat, but at the same time, from the same area, we managed to produce more biomass, and we didn't monitor any problem in terms of disease and weeds. Because if you rotate the crops, you can help them to clean the fields and to reduce the chemicals, the herbicides. Here you can see in this photo sun hemp, that is a leguminosa, fabaceae crop. It is a biomass crop, but at the same time, it improves a lot the fertility of the soil. It can think as a leguminosa. We have fiber, sorghum, kenaf, and hemp. If we go to the next slide, we can see just a view of the field where we rotate wheat with sun hemp, industrial hemp, kenaf and wheat. And now we are in the 5th year of the project, and we are going to complete 2 rotation cycles in a period of 7-8 years. And we do believe that we are going to have sound results in terms even on biodiversity. So if we go to next. Next is, you know this crop very well, even in Lithuania. In industrial hemp is very important crop. You can imagine how big, we have a large variety of end uses, and it is a crop that it has been, it cleans definitely the crop, the field from the weeds. So if we follow wheat after hemp, we can reduce a lot the herbicides needed. Let's go to the next. And here again some photos from perennial grass just to see how the crop it looks. And it is considered when we are going to remove the field from the crop, the crop from the field is about 15 or 20 years. The soil carbon definitely is going to be improved. And what we are doing right now, we try to prepare some common articles in the place of MAGIC project, where we're testing also this crop to prove this, how the soil fertility of the crop is going to be increased after the removal of miscanthus. And let's go to the next. Lithuania has a great interest on willow and poplar, 2 very important woody species cultivated for biomass, bioenergy, and the area of the cultivation in Europe on this, it's about 20,000 hectares each. It's not so it's not small, but is increasing gradually. And both of crops, they consider environmental friendly and friendly in terms of biodiversity. These crops we used to grow to harvest every 3 to 5 years to take the biomass. So let's go to the next. Here in this slide, that is my last, I want to provide to present some useful links to get information on non-food crops. So the first link is on Panacea platform. Here we have 29 non-food crops. You can find information, and now we have uploaded the country reports from 10 European countries like Lithuania, Poland, Italy, Greece, etc. You can see which are the top 5 non-food crops for their country. And if I go through all of these countries, I can say that hemp, camelina, miscanthus, willow and poplar, and maybe sorghum are the top 6 non-food crops for Europe, that they are real near to agricultural practice, and they can be grown in rotation or can be grown on marginal land. And then we have 3 places to visit the MAGIC project. You can find a database in the first page, the MAGIC maps, where you can find where the marginal land located in Europe, and a decision supporting system for the farmers. Thank you very much for your attention. Once again, thank you very much for the invitation.
And thank you very much for a lot of information and a lot of knowledge you shared through the projects you have delivered and still working on. While I was listening to you, and I just wonder, because non-food crops is slightly different, than obviously not slightly, a lot of different from food crops. And I wonder what role gardeners play in growing non-food crops in their gardens and actually helping with the biodiversity, helping with improving the soil conditions as well. Is there, the research that you have done is on the big scale, and I wonder what collectively gardeners, people growing non-food crops in their back gardens could do to collectively improve the biodiversity.
For sure, but there they will be just to increase the biodiversity and not to produce feedstock for bio-based material and bioenergy. In our case, we want to offer to the farmers an alternative to increase their income, and if possible, to avoid monoculture, to increase biodiversity, to have healthier soils, and maybe an opportunity to grow the marginal land that left abandoned. And in this way, with low indirect land use change to produce an extra income. Of course, always not decrease the biodiversity. And keep in mind that in the new CAP, I told you before, all these issues will be very important and emphasized.
Yes, they will be important and emphasized. And I wonder how it's all through your project. Maybe you can share an example. You reach those farmers, you know, who might be wondering, because not everybody has access to the same information. I wonder what tools and techniques you have used.
In Panacea, each partner tried to make a lot of events locally. So each partner like you, like me, try to do at least 3-4 national workshops and try to attract the Union of Farmers, Union Farmers Cooperatives, to present what we are doing and to show how they can grow this crop. And finally, we prepared training material that presented in the training of the farmers, of the students, of the agronomists. So we try to be a tool for them.
And I suppose it really kind of influences and adds to it creates a value added to what the European Union as a union is trying, and European Commission is trying to achieve, isn't it? To move towards a zero pollution.
Yes, always. This is the main target, and of course, always thinking also to the farmer and how we can help them, but of course, not to harm the environment and not to destroy biodiversity, because we have to combine all of these elements in order to be successful.
Yeah, absolutely. And how do you work with the scientists who actually are very pro food crop and say, no, actually we need more land to develop new crops to feed the nations, whereas you're saying, how are you working in collaborations? Because you're saying, no, we need biodiversity, we need this.
But in the example of Bikul, that were between wheat and corn, we put in the middle that the land was fallow. We put other crops. This is a good example that between of the main crops, we introduced non-food crops to increase the biomass and biodiversity.
Yeah.
So and increase finally the income of the farmers.
Yeah. So it has to be both ways, isn't it?
Not to replace, just to give another plan. This is very evident also in France when they promoting the cover crops, the catch crop, the double cropping, not to remove corn and wheat, but to grow in a way that even non-food can fit.
And this is, I think, a very powerful message, isn't it? That they can grow in collaboration and two different aims can be achieved as well.
Yes. But the farmers they want information and how to do that.
Yeah, absolutely. So I think it's a job for the scientists and the networks that they're involved in to spread this knowledge and information. Effie, thank you so much for your presentation insights. I can see Vita has shared the link to the Panacea platform that we can access and look at the data and the maps. I'm sure there's going to be a few questions. I hope you can stay until the end of the session. So if there's any questions, you can answer them in the chat.
And last but not least, I would like to congratulate Vita because she did an excellent, and her team, an excellent job in Panacea. So active. If you go through, you can see the video that they produced and so active in the social media.
Well, it's lovely. It's a pleasure to hear that. I think so. I know Lithuanians are great anyway. Just saying as a fact, as well as everybody else.
Thank you very much.
Thank you very much.
Stay and I will check for questions.
Brilliant. Thank you very much. I'm sure there will be plenty. Thank you. And now I would like to invite Dr. Andrus Menikis, who works at the Department of Forestry, Mycology and Plant Pathology at Swedish University of Agriculture and Science. So from Greece, we're moving all the way to Sweden. And Andrus has a research interest in biology and control of invasive tree pathogens, population genetics of fungi, and many other areas. And today we're going to hear some insights into the future of the forest. Andrus, the floor is yours.
Okay, thank you very much for this nice introduction. And I would like to thank all organizers for this opportunity to be a part of this event and to present our project, which is conducted in collaboration with researchers at the Lithuanian Research Centre of Agriculture and Forestry. So you said our interest is looking into these challenges of climate change and diseases which we face today. And we want also to look beyond the current knowledge and look into the future, what possibilities we have to sustain functioning of forest ecosystems. So in this presentation, I will try briefly to remind all of us what are the current situation with forest damages, including both biotic and abiotic forest damages in Europe. And in the second part, I will just briefly outline the idea of our project and main activities. So just to start with biotic forest damages, I would like to show that there is increasing number of different reports and publications across Europe that the different pathogens, especially new and invasive pathogens, get established here. And these pathogens and different pests affect different tree species. They occur in different countries. And also these pests and diseases, they involve different organisms, such as insects, fungi, nematodes, and bacteria. And if we look at the European map, we see a different situation in different regions, and we can see that Southern Europe is most affected, and Northern Europe has a more mild effect. For example, Lithuania has currently quite mild effect, but I believe this situation can change rapidly, and we need to be alert. And this basically is driven by climate change and globalization, especially by the transportation of different products and goods, and so how these different pests and diseases spread across different countries and even continents. So if we summarize all this information, we can see this graph from the past, and we can also predict where we look at in the future. So the situation is not good. So we see this gradual increase of these invasive pests and diseases in Europe. So we can also expect that the damages will also increase in the future, and we need to be prepared for that. So apart from the biotic forest damages, we also face our climate change, and which is basically this abiotic forest damages caused by climate change and different factors. And some examples are this, for example, temperature anomaly, which we had in Sweden and overall in Scandinavia in 2018, which was very high temperatures and during very long period of time, which in fact resulted in very large areas of forest affected by the drought. And in addition to that, these high temperatures and heat caused very extensive fires. So in principle, it was the most extensive forest fires in modern history. And this caused about 25,000 hectares of forest burned, which can translate to about 100 million euros of forest loss. Besides, we talk today about the carbon footprint. So you can only imagine how negatively it contributed to the carbon release to the atmosphere. So both biotic and abiotic forest damages, they both negatively contributed to carbon footprint. Apart from forest fires and drought, we also have this wind damage to forest, and especially storms have a major impact on forest, and forest especially which is planted in proper areas, like our spruce planted on pine signs, would be very heavily affected. As you can see from these pictures, the damaged forest after the storm, and the very large accumulation of food as a result of the storm. And in addition to damage, this would also provoke incidents of insects, like bark beetles, which can continue damaging the remaining forest. So we see this interconnection of different agents of damage. And again, just to summarize this, what we see and what we can predict, we can predict that these damages can increase in Europe, partly because of climate change. And again, we need to be prepared and be aware. So the general idea of the project is just to collect and have a better understanding on how climate change and tree diseases may affect the geographical distribution and health of forest trees and associated biodiversity. And we can have this classical triangle when we have biotic factors, and forest trees, and look at how these factors interact among each other and what will be the outcome of this interaction under different scenarios of climate change. The aims of our project is to study possible impact of climate change on forest health and on microorganisms, which constitute one of the principal components in forest biodiversity. But we also are interested in different components of biodiversity, such as like insects and fungi and lichens, and how these individual components interact with host trees. And finally, we want to look beyond the current knowledge and see if something happens, something extreme happens, and the local tree species cannot survive and support the local biodiversity. Can we use some alternatives? Can we use non-native tree species as alternative hosts to support the local biodiversity? We are not advertising planting with non-native tree species on a large scale, but I believe we need to be open on for different options and opportunities, especially for future. And in this project, we use a number of different methods and like a field sampling and observations and climate data, and we do some laboratory analysis, for example, fungal culturing. But in principle, the project is DNA-based, and we use DNA metabarcoding as a method using ITS as a marker in combination with high throughput amplicon sequencing and bioinformatics and statistical analysis. So now I will briefly go through activities in this project, and in total we have 5 activities. So I just will not go deep into the results, only just give you an overview. So in the first activity, our interest is the biodiversity and function of microorganisms associated with Scots pine and Norway spruce under different forest health conditions. And in this activity, we have this a number of research plots across Lithuania for both for pine and for spruce. And for each plot, we have silvicultural data, and we also collected the forest health condition data. And then we try to link these data with microbial communities and just possibly see the effect of these communities on health of a forest. For example, just what this graph shows how different sites can have different health status in Scots pine forest, and we see that different parameters quite well correlate, which is logical. So as I mentioned before, we do field sampling, and we sample different materials such as needles and twigs and soil and roots, and then we process these materials in the lab and basically do high throughput sequencing, which generates a large number of these microbial species. And as I said before, we try to interlink these communities with forest health and see what the prospects we can have for the future. In the next activity, we try to model the possible impact of climate change and of tree diseases on geographical distribution and composition of future forest stands and associated biodiversity. And for this reason, we have a transect which expands from the along this north-south transect of pine and spruce distribution in Europe. So we use a similar approach when we collect different materials and evaluate the health status in different stands at the different climatic zones. And just to be able to predict how any change in the climate can also affect these forest and microbial communities associated with these trees. Just very briefly to show that, for example, this is for spruce, what we can see now that one is like in the south and 12 is in the north, and we see that forest condition deteriorates in the north, and what the reason for that is not known, but we will, during our studies, we try to uncover this question. Okay, and the next activity, or this activity, is devoted to the ash trees and Fraxinus excelsior in particular, which is heavily affected by invasive pathogen across the whole continent. And our interest is looking into the biodiversity impact of different microorganisms on health of these trees and how these different microorganisms can suppress the pathogen itself. So in different parts of Lithuania, we have a number of research plots, and we collect materials from both healthy looking and diseased trees, and we process these samples in laboratory by using like classical fungal culturing methods, and also we deal with like a large number of different species, and our aim is just to using this in vitro test just to screen for their properties, just to see how different isolated fungi interact with fungal pathogen, and if any of these can be used to suppress this pathogen. In addition to that, we also apply this DNA metabarcoding approach to study fungal communities associated with functional tissues on soil of healthy and diseased ash trees. We know what this process is primarily driven by the genetic factors and by environmental factors, but I believe little is known how associated microbial communities can also affect this pathogenicity process, so we look more deep into that. In the next two activities, we try to look into the different tree species as a possible alternative to local tree species, and how good or bad they can be to supporting local biodiversity. And in this fourth activity, we'll look at the potential of large trees as an alternative host to soil and wood inhabiting fungi, lichens, and insects of Norway spruce. So the principal question is, can large trees can be planted on a large scale without compromising biodiversity currently associated with Norway spruce? We know currently that the larch is planted in Lithuania, but only on a limited scale. And again, we have a number of research plots, and we do different measurements and collect different observations for insects and for fungi and for lichens, and try to evaluate this biodiversity specifically associated with each tree species and the different health condition, both on the living trees and on the dead trees. And just very preliminary, some observations, we see that there is quite a good overlap of species associated with both tree species. But when we look at in this multi-dimensional analysis, we see that there is some specificity that, for example, living large and living spruce trees share at least part of the communities, but dead spruce and the large trees have quite different insect communities. So this is important to take into account. And the next activity is devoted to the hardwood noble trees like our ash and elm, which is currently affected by this invasive pathogens. And in this activity, we follow a similar approach when we collect field materials, and again we do this comparison among the local and non-native tree species, and basically just to understand how good are they in supporting this local biodiversity. So with this slide, I would like to end this presentation, and I would like to thank the Lithuanian Research Centre for Agriculture and Forestry for hosting this project. And I would also like to thank Research Council of Lithuania for providing funding. And also I thank Research Group. And thank you all for your attention.
Thank you very much, Audrius. The research they're doing clearly is looking, as all the research we've seen today, towards the future and how we can sustain it. And I wonder, just a thought while you were talking, if there's going to be more research done on why forests are deteriorating in the north faster than in the south, and there's more diseases, as you mentioned. Are there any planned research, or do you have more details? It's quite interesting, and I wonder if it's related to the climate change, that the climate change, the effects of it are noticeable more in the north forest rather than the south.
Yeah, I believe so. The climate change has a major impact, especially on these areas, and it can be so that it promotes, establishment and spread of new diseases, and we have some indications on that. So, yeah, I believe so.
You're talking about the different pathogens and diseases that already exist in our forest. Is it actually possible to sustain it? Or is the climate change moving too fast for the scientists to catch up with the technology and do the research in order to, I don't think, can it be stopped even?
I believe so, at least to some extent. And I believe that it needs to be knowledge and research based, these decisions. So basically our project is just to provide some basic information to decision makers, just to be able to make wise decisions.
And following that, obviously, so as I understand, then you will be working, you will inform policymakers in order to change some of the policies on the national and probably international level too.
Yes, I believe so.
Thank you very much. I just want to remind all the presenters to keep an eye on the chat, because I can see there's some movement and some questions being posted. So if all the presenters can check the chat and get back to those who asked the question, that would be great. And now I would like to ask and invite our last presenter of this session, Dr. Vaida Sirgedaitė-Čežienė, who is a head of Forest Plant Biotechnology Lab and chief researcher as well. And she has an interest in forest phytocynosis, assessment of forest vegetation, climate change, and others. Vaida, the floor is yours.
Yes, hello. Hello to all participants and organizers of the conference, and thank you for opportunity to present shortly about, as I don't know.
Can you share the screen?
Yes, and do you see the presentation?
It's just loading. I think it's going to be up in a few seconds. Yes, we can see that. Thank you.
Okay. Okay. And I want to present shortly about biotechnological research and exploitation of various physical, chemical, and biological agents for the improvement of plant productivity. Negative consequences of the chemical preparation applications are known to include pollution of the environment, as well as of agriculture and forest productions with residual chemical preparations such as heavy metals, nitrates, and other compounds hazardous to the health. The basic tendency in the field of plant protection concerns with reducing the use of pesticides and their replacement by environmental acceptable biological preparations. And I want to present fields of research which are carried out in laboratory of forest plant biotechnology during long-term studies, which can lead to achieve zero pollution. Physical, chemical, and biological tools such as cold plasma, electromagnetic field, hydrogen, microbiological research of plant-associated bacteria and fungi can be used as a tool to reduce environmental pollution in different ways. In vitro studies are carried out with many tree species in our laboratory, but popular species and its hybrids are used as a model tree for their quick growth. Also because it's a model woody plant for the microorganisms and plant interaction. Some other plants species used for the in vitro studies in our laboratory are known for their nutritional or decorative value, for example, rosa or stevia. To determine the multiple effects on plant biochemical and morphological parameters during experiments, the laboratory analysis carried out. It is important to determine the possible positive or negative effect to the plant response during primary growth stage, which can affect a long-term growth. For that reason, I pointed some biochemical analysis and morphological analysis which are carried out in the laboratory during experiments. For example, assessment of enzymatic antioxidants, oxidative stress damage level, antioxidant activity, quantification of secondary metabolites, nutrients, proteins, photosynthetic pigments, and some morphological parameters, for example, shoot growth, root system development, or flowering. And now I want to present some our tools used during experiments. Plant changes their physiological or biochemical parameters when they get signals which are induced by various stressors. These changes are related to selective modulation of protein expression in the growing seedlings. In recent years, much attention has paid to the positive effect of innovative technologies such as low temperature plasma and electromagnetic field treatment of biological objectives, giving rise to a new scientific field, plasma biology. These technologies are eco-friendly, safe, and they can activate seed germination, but do not cause genetic mutations at the same time. Several reviews summarize the findings of the laboratories around the world, indicating that seed treatment with physical stresses can promote seed germination, stimulate plant biochemical processes, increase biomass gain and yield. In addition, treatment with physical stresses has been shown to be effective in seed microbiological decontamination and lead to increasement plant resistance to pathogen. And here you can see the experiment of our pilot study. Forest tree breeding in Lithuania is based on half-zip family test or field trails. Therefore, the genetic selection is one of the most important factors during research. Studies showed that the height of growing seedlings of several genetic families was increased after seed treatment with physical stressors. This may be related to the ability of plants to synthesize more photosynthetic pigments, which are associated with growth potential and productivity. Seed treatment with physical stressors increased secondary metabolite content in seedlings. Moreover, significant correlation have been determined between secondary metabolite synthesis and the resistance to the pathogen. Chitin and its derivative chitosan have been known worldwide for its potential as a biocontrol agent. Due to biological and chemical properties, for example, biodegradability, non-toxicity, biocompatibility, and fungicidal effect, the chitosan has a broad range of industrial applications. In medical science, chitin has been used as a wound dressing material, drug carrier, and anti-cancer drug, while in chemical science, it has been used to prepare an affinity chromatography column and to immobilize enzyme. It has been used as a food additive and biopesticides in the agriculture industries. In cooperation with business companies, we develop a novel chitosan-based product from insect Tenebrio molitor waste for promotion of plant growth and resistance. Why from insects? The skeleton of insects contain 45% of chitin versus 90% for the shrimp shells. It contains lower amount of calcium carbonate. Therefore, according to these aspects, chitin can be more easily extracted, resulting in a more environmentally friendly processes. A project just started, and we expecting interesting and innovative results. Most plants are naturally colonized with symbiotic bacteria. Plant symbiotic bacteria may be beneficial to their host both directly and indirectly through a variety of mechanisms that result in plant growth promotion and resistance to pathogen and different stressors. It can be also used as biofertilizers. Biofertilizers usually consist of microorganisms that bring about an improvement of the nutritional status of the soil by enhancing nutrient uptake and accessibility to crop plants, or by producing secondary metabolites that affect plant in a positive manner. A potentially symbiotic bacterium was observed in the tissue culture of hybrid aspen. It was isolated, and a colony of tested bacterium was grown separately on the LB medium. The studied bacterium was identified using DNR methods. A successful example of cooperation with business companies is the bacteria-based product. The product for plant growth promotion developed from bacterium which was isolated and investigated in our laboratory. The tested plant species, for example, rose species, were positively affected by bacterium Penibacillus both in vitro and ex vitro. The further research were carried out with oaks. because of their extensive habitat at long and long lifespans, which are valuable niche for the various beneficial bacteria. Isolated bacteria strains from oak leaf endosphere were tested for several traits beneficial to plant growth promotion. For example, nitrogen fixation, phosphate solubilization, mineralization, production of iron transport agent siderophore, and production of plant growth regulator. We have tested the effect some of these bacteria on model populus species microshoot growth parameters in vitro. As you can see, a positive effect were observed on root development parameters after inoculation with bacterium Penibacillus. We can consume that presented physical, chemical, and biological patterns would be beneficial as a tool for agriculture and forestry management. Selected and developed novel products and processes could be recommended in order to effectively activate plant growth, to increase agriculture and forest productivity, to reduce environmental pollution caused by chemical pesticides and pathogen damages in the future. So thank you for your attention.
Thank you very much, Vaida, for sharing best practices, how to work successfully with the businesses. And I think so that is quite inspiring. So I just wonder if you're planning to work on any other bioproducts and with the businesses and maybe using the new technologies and maybe take them to the market.
Yes, of course. Well, all the time we're looking some interesting ideas that it's not so common to our carried out projects. So I think, yeah, yes, in the future, yes, we will.
And are those the collaborations that you're having with the businesses you said, are they Lithuanian wide or are you going out to the Europe as well?
At this time, Lithuanian companies.
Okay, but I think what we're saying here as well, that if there are any potential collaborations outside of Lithuanian borders, you know, you would be very welcome to receive those and maybe use some innovative technologies.
Yes, of course.
And
it will be very interesting.
Yes, and you talked about the experiment you had with the oak, English oak. That's correct,
yes.
Right? And you used
yes.
one of your products, as I understand. Could you elaborate a bit?
No. With oak only just a pilot studies, just a start. So, but we're expecting very interesting and promising results in the future.
Okay, and do you think that can be applicable to the kind of bigger scale as well? So not only apply to the oak tree, but other species too?
Yes, sure.
I just wonder how it links with Audra's presentation that they were talking about the forest and looking at genetics and the DNA. So I just wonder if there's some sort of an overlap as well that could happen in the future.
Maybe, maybe.
Good. Thank you very much, Vaida, for your questions. And thank you all of the presenters and scientists who have shared their vision and insights and projects that I'm sure are going to make an impact, because one step at a time that will make a change. We have finished with the second session of today on biodiversity of the crops in the forestry and agriculture. And we have seen that it's incredibly important to be diverse and take different elements into account. Thank you for the active discussion in the chat. And if there are no further questions at this point, we're going to break for a slightly longer break because we had a short in the morning, and we will see you at 2 o'clock, yes, at Central European time. So I would like to wish you a good break, and we'll see you in 25 minutes.